Assalamu alaikum. My name is Aqsa Arshad, student of MS Zoology from Department of Zoology, University of the Punjab, Lahore. The topic of my presentation is reprotoxicity and abnormal steroidogenesis following oral exposure of tartrazine in mice and remedial potential of Allium cepa. The brief introduction of this dye is it is a synthetic food color, imparts yellow color in various food items and belong to the class of azo dye compounds. It's a high consumption in various dietary items such as biscuits, sauces, noodles, and sweet meat were reported. However, this dye is nowadays highly consumed by children in junk foods and is responsible for various hypersensitivity problems and behavioral changes in children. In addition, noxious consequences are also reported on reproductive system in mice as well as steroidogenesis in both mice and animal models. The objectives of my study were as this dye is nowadays consumed above their ADI levels, especially by the children and adults. So there is still ambiguity that exists regarding its reproductive toxicity. So present study is executed to explore the reproductive toxicity of this dye, to explore abnormal hormonal functions in mice model, as well as the use of aqueous onion extract as a protective source against the steroidogenesis and reproductive toxicity caused by this dye. Moving towards the materials and methodology, both genders, male and female mice, were bred and maintained in the Animal House of Zoology department, and their required doses were prepared as reported by the literature, that is 0.140 milligram of tartrazine and zero point 0.1 ml of aqueous onion extract. The animal study design was designed in such a way that both male and female mice weighing 14 plus minus 1 gram were selected, 30 male mice and 30 female mice, and both male and female mice were then divided into three groups as control group, which was administered along with water. Second one is dose group, which were given 0.140 milligram of tartrazine, and the third one is dose plus antidote group, which were given 0.140 milligram of tartrazine along with zero point 0.1 ml of aqueous onion extract. Both genders were given 30 days of treatment period, and after that they were sacrificed according to the protocol provided by Ethical Committee of Punjab University, and the following parameters were assessed such as body weight, gonadal weight, biochemical analysis. In addition, histopathological defects of testes and ovarian tissues were also examined. Then next is results of all above parameters. First of all is body weight of male mice, which you can see in this graph that it is decreased, significantly decreased in dose group as compared to the control. The next one is the body weight of female mice, which also showed a significant decline in the dose group as compared to the control. However, you can see in this graph that when dose is administered along with the antidote, which is the aqueous onion extract, the female body weights were resumed to some extent. The next one is organ weight of male mice, which is testis weight, which is also decreased significantly in the dose group as compared to the control. Organ weight of female mice were also showed a remarkable significant decrease in the dose group as compared to the control. But if we compare the organ weights of both male and female mice, you can see that testis weight were decreased in dose group and ovarian weight were also decreased in dose group in the case of both male and female mice. But you can see in this graph that the organ weight of female mice, that is ovary weight, showed a more remarkable or a significant decrease as compared to the organ weight of male mice, which shows that this dye poses more noxious or consequential effects on the female reproductive system as compared to the male reproductive system. Then moving towards the male reproductive hormone analysis, which is the testosterone, which is the main reproductive hormone in males. It shows a significant decrease in dose group as compared to the control in terms of mean plus minus standard error mean. Then the semen analysis of male reproductive system were examined, which includes the sperm count in terms of millions per ml, normal sperm number and abnormal sperm number. The sperm count in the male mice were decreased in the dose group as they are treated with the tartrazine dye. Next, the normal sperm numbers were also decreased in dose group. When, however, you can see that when dose group was given, mice were treated with dose plus antidote group, their normal sperm number were resumed up to some extent and was somehow near to the control level. In the case of abnormal sperm number, the abnormal sperm number were significantly or remarkably increased in the case of dose group when compared with the control. So male reproductive hormonal analysis, testosterone and its semen analysis, sperm count and abnormal sperms were significantly increased in those groups when compared with the control. Then moving towards the female reproductive hormonal analysis, follicle stimulating hormone level were decreased in 4 weeks old mice as shown by the different superscripts. Luteinizing hormone in female mice when treated with the dose group, that is tartrazine, also showed a significant decline as compared to the control. However, when female mice were given dose along with the antidote, means dose plus antidote group, the luteinizing hormonal level was almost similar to or near to the control level, which means that Allium cepa has some sort of antioxidant capacity in resuming the level of luteinizing hormone in females. Then moving towards the estradiol level, it was also showed a decline in the dose group when compared with the control. Then moving towards the histology, first of all, histology of testes. As you can see in these slides, the picture A is of control group, picture B of dose group, and picture C is of the dose plus antidote group. As you can see in control group slide, the normal organization pattern of all the reproductive cells of males, that is lumen, properly assembled lumen, spermatozoa, Leydig cells, and and the spermatogonia are can be seen in the control group of male mice, which shows the normal organization of all these cells. While in those groups, you can see that it shows a reduction in the number of spermatozoa. You can see that in the seminiferous tubules, the number of spermatozoa are declined. On the other hand, it also shows the dilation of the seminiferous tubules, and interstitial species also increases. In the case of dose plus antidote group, it shows the some restoration of lumen with the sperm number shown by the red bar, which somehow increase, means lumen is again organized and the sperm number increased to the some extent. However, spermatogonia and spermatocyte cells can also be seen in dose plus antidote group. However, in the case of Leydig cells, they still show the degeneration or reduction in their number, as represented by this blue arrow. And these testis slides were observed at 10x using the H&E stain. Then moving towards the histology of ovaries, both A picture is of control group, B picture is of the dose group, and C picture is of the dose plus antidote group. As you can see in control group, there is a normal development of oocytes, secondary follicles, antral follicles, properly organized corpus luteum and follicular cells can easily be seen. However, in the case of dose group, you can see that follicle atresia is occurred, as shown by the yellow star. Also, the vacuolations were seen in the case of dose group in the female ovarian structure. Then moving towards the dose plus antidote group, which is a C picture, it shows that increase in the number of follicular cells, especially the primary follicle number is increased, secondary follicle number is increased, as shown by the yellow and red triangles, which shows that somehow the female follicular cells and ovarian structure was resumed when dose was given along with the antidote group. So the conclusion of my above work was that tartrazine has damaging effects on reproductive parameters and also induced the some testicular and ovarian tissue damages. Also showed a decline in the levels of male and female hormonal levels such as testosterone level, follicle stimulating hormone level, estradiol level, and luteinizing hormonal level. However, we can see in all these slides that when those were administered along with the antidote group, somehow all above parameters such as body weights, organ weights, and hormonal defects were resumed to some extent, which means that Allium cepa, which is aqueous onion extract, was somehow beneficial or show its potential in resuming all these factors to its normal values or near to the control values. So, however, keeping in view all above toxicological problems resulted by this dye, it would be better that this azo dye would be replaced by some coloring compounds that contains the natural ingredients. Thank you.